last part of this is now about, with all of this stuff that we know about the cell cycle and how to regulate it, we can also take cells out of the body, culture them, which means to replicate them in an external environment. It's got a lot of really, really cool purposes, particularly for studying things like trialing vaccines, or how exposure to certain things will influence certain types of cells and how it will change gene expression and things like that. So there's heaps and heaps of applications of taking cells from a living organism and artificially or exposing them to a culturing in a lab environment where they replicate, and so they continue to live and thrive in that kind of environment. Have a watch of this video here and have a look at the next video on the next slides as well. But essentially, we culture for a variety of purposes. So to be able to be successful here, you want to be able to explain, in essence, the three things or three steps of how we culture. And then you want to look at potentially what are the key requirements for being able to culture. Make sure you do go onto this presentation and watch the video. As always, they're there for a reason. But in essence, culturing as a process is broken down into three steps. The first part is the dissecting step, where we need to take the tissue or the cells of interest. So say we're looking at being able to culture skin cells, we need to take a sample. So we dissect it to expose the cells of the same type. So we get a sample of skin cells and we then get them ready. What we then do is we do a process called suspension. And suspension has a variety of sort of key importance reasons, but really what we're trying to do is get it ready for further mitotic division. And so if we're looking at, say, skin cells, for example, they're all stuck together at the moment. And so when we suspend them, what we are trying to do is get those skin cells to break apart, because we know that, and I'll go back here, we know that there are some factors that affect cell division. And if we look here, you can see density dependence was one of the factors that influence cell division. And so if we want to be able to get cells to continue to grow and divide, if we're talking about skin cells and they're already all in contact with each other, we're not going to actually get the continued growth of those cells once we take them out of the body. So we need to be able to separate them. And that's the purpose of, one of the purposes of suspension, is to be able to do that, is to be able to isolate the cells from each other so that then we can actually go, all right, now you're free to grow. And the last part in this, the culturing part, is really, really the most important. And the next slide has all of the things that we need to be able to have successful culturing or successful rounds of mitosis actually occurring in a lab environment. So we get the cells of interest, so the skin cells that we're after that have been separated, and we place them on a suitable culture medium, so like an agar plate. We then have to provide the conditions required for mitosis to occur. And so those conditions look like the right types of temperatures, sterile conditions, the optimal pH. We're talking about all those things like what we need for enzymes to be able to do their jobs effectively. We need nutrients, we need sugars, amino acids, we need all of the things that our body naturally needs to be able to grow and divide. So all of these are much the same that we need in a lab environment. We also need access to oxygen so that we can conduct aerobic respiration, because we want to be able to have those cells being able to catalyze and break down like glucose to be able to produce energy so that mitosis can therefore occur. And so you got to think about all of these conditions being much the same as the environments that are favorable inside of our bodies. We're mimicking that in a lab environment so that we can then get those cells to grow and divide. But why? So it's much, much more ethical to be able to do things like test pharmaceuticals or cosmetics, or test the influence of vaccines and stuff like that on something that is not, technically living or belonging to part of an organism at that point in time. So we would do these processes so that we can actually make sure that we advance as a society, so that our scientific advancement can keep going. We might be able to cultivate skin cells for using in grafting and stuff like that. So there's a lot of applications there. We might be able to continue protein production, so getting desirable proteins being created in an artificial environment, cloning certain cells to replace damaged cells. There's heaps and heaps of applications of this. But this only works, remember, if conditions are met and if we can do those